Hello friends, this is the 6th episode of the channel Ace Botany, dealing with +1, +2 botany chapters, useful for theory as well as NEET model exams. So already 5 episodes over. So if you watched it, if you like it, please share, subscribe all, comment and like it. Now we can enter directly to the topic. So we can start. Today we'll be dealing with the male reproductive part of a plant. So for that, I will share the screen. So it'll be easy for you to get the diagram and all the things. Now screen will be visible to you. So we can proceed. So male reproductive structures, which include stamen. After that, we will be entering into microsporangium, pollen grains, male gametes. Initial part is called microsporogenesis. Genesis means formation. Microspore or male spore will be formed. So we can start with a typical stamen male reproductive structure in plants, which has got 3 parts: anther, filament, and connective. Anther will be at the top part of the filament or free end. Anther will be attached. Bottom part, that is called proximal end. Proximal means near to the point of attachment. So base of the stamen will be attached with the thalamus. So 3 parts will be there for a typical stamen: anther, filament, and connective. Now a typical anther is, that is our popular question, it is a bilobed structure. 2 lobe wing will be there, and each lobe has got 2 theca. Theca means chamber. So each lobe 2 theca, 2 lobes, so total 4 theca. So the term is tetrathecus. So a typical anther is bilobed and tetrathecus. Each lobe of the anther contains 2 theca. So I think that particle is a very predominant question. Exceptions are there. Even though not in NCERT it is mentioned, but family-wise it can be a question. Always you have to learn. Monadelphous monothecus reniform anther is seen in Malvaceae. Now, when we look on the diagram, you can find there is a term or a structure called connective. So what is originally this connective? Connective is a deep groove. It is a deep groove and it is attached by a band of vasculated. Next term is important, sterile tissue. So vasculated, sterile tissue. Connective is a deep groove attached by a band of vasculated sterile tissue. Now, I think this page is clear. Sometimes it will be a labeling question, specifically the first one or the second NCERT diagrams. I think it is clear. So we can proceed to next stage. When we proceed with the anther, we have to start with the anther structure. Then we will be studying microsporogenesis. So different layers can be seen, and that is again a theory entrance question, including a TS to draw or LS to draw and represent the parts. Label diagram both theory and entrance. Now, first layer, outer layer is called epidermis. Epi means above, dermis means layer. So normally it will be single layered. Since it is epidermis, function will be protection. Second layer, it is called endothecium. Endothecium is below epidermis, and this layer has to do a major function, and that will help dehiscence of anther. Dehiscence means split open. And how this anther gets split open? By hygroscopic action. So what is meant by hygroscopic action? Taking water in and out. And which chemical will be doing it? It is done by alpha cellulosic fibrous band. It is a question even though not directly mentioned in NCERT. So due to presence of this alpha cellulosic fibrous bands, and that is connected with the inner tangential wall of the anther, inner tangential wall. And that will be playing hygroscopic action. by which helps in dehiscence of anther. That is endothecium layer. Third layer is middle layers. Generally, it will be 1 to 3 layers. Middle layers. It is ephemeral. Ephemeral, we already discussed. Ephemeral means short-lived. Later, that layers will undergo degeneration. That is why it is short-lived. Ephemeral. Usually 1 to 3 layer middle layers. Fourth one, that is tapetum. Tapetum is the innermost layer, and which usually surrounding the sporogenous tissue. Main function of tapetum, a common question, is nutrition of the developing pollen grain. So which will be the nutritive layer? In anther, it is tapetum. And this tapetal cells has got many abnormal features. Wedge shape, multinucleate, DNA content will be variable. That way, many abnormal features will be there for tapetum. So this wall layer is little bit important, either to diagram-wise labeling or entrance question. So layers will be epidermis, endothecium, middle layers, and tapetum. Inside the tapetum, we can find microspores. So how these microspores as well as pollen grains will be formed? So again, we are entering into a topic, microsporogenesis. I represented a diagram. You can find it. A young anther in tears shows a mass of diploid cell surrounded by an epidermis. Somewhat rectangular, that way we can say, or irregular shape. As the development proceeds, it gradually comes to a rectangular shape. And later it becomes a bilobed nature. So third diagram, if you observe, we can find it. Now, in this bilobed structure, from 4 corners of the anther, 4 cells become predominant. And these cells are called archisporial cell. This archisporial cell, each one will undergo transverse division, forming outer parietal layer and inner sporogenous layer. From parietal layer, we can find wall layers will be formed. Sporogenous layer that will be producing microspores after meiosis. This is how it takes place. So mass of cell attaining a rectangular shape, then become bilobe, 4 corners, 4 cells become prominent archisporial cell, undergo a transverse division, outer parietal, inner sporogenous. Parietal cell will give rise to wall layers. Sporogenous cell will give rise to microspores after meiosis. Now, each microsporangium will be located on 4 corners of anther. And inside this microsporangium, you can find microspore mother cells, which will be occupying center of the microsporangium. So position is important. Microsporangium, 4 corners of anther. Then microspore mother cell in each microsporangium will be at the center. This is how microspores will be formed. That part, I think it is clear. Now we can proceed to next area. Microspore mother cell, again undergo meiosis. 4 haploid microspores will be formed. Microspore mother cell, otherwise called pollen mother cell. And before the anther matures, dehydration will be there. And 4 microspores formed after meiosis will dissociate. Anthers will dehiscence and followed by releasing pollen grain. So once again, microspore mother cell undergo meiosis, 4 haploid microspores will be formed. Pollen mother cell or microspore mother cell. From that only, these microspores will be formed. So before it, when anther matures, dehydration will take place and the formed microspores will be dissociating and further dehiscence of anther followed by releasing of pollen grain. This is how microsporogenesis takes place. 4 microspores will be formed and they have different types of arrangements. Most common is tetrahedral tetrad. So 4 microspores in a group, so it is called a tetrad. Tetrahedral tetrad will be most common arrangement. Various other arrangements are there, but not in our direct syllabus. So this is how microspores will be formed. Now we have to proceed to pollen grains. Microspores, again on further development, converting into pollen grains. Pollen grains are supposed to be the male gamete of 5. And size of the pollen grain, according to size of the pollen grain or your microspore, we can say 25 to 50 micrometers in diameter. 25 to 50 micrometers in diameter. Shape will be for microspores, usually spherical. And palynology deals with study of pollen grains. So 3 aspects: microspores, pollen grains, male gametophyte, size 25 to 50 micrometer, shape will be usually spherical, and palynology deals with study of pollen grains. There are different branches for palynology. A typical pollen grain has got a layer called sporoderm, and sporoderm will have 2 separate layers, exine and indine. Another important question. What are the properties of exine? What are the properties of indine? And how exine is commercially utilized? So sporoderm, outer layer is exine, inner layer is indine. Exine is made up of a specific chemical called sporopollenin that is supposed to be one of the most resistant chemical of this world. And that is one of the reasons why these pollen grains become fossils. And on this exine, beautiful designs will be there. And now in ornament fields, these designs will be analyzed. Photographs will be taken. Based on it, ornaments will be designed. So a new branch is developed. It is called exine sculpturing or ornamentation. And on this exine, we can find certain openings. Openings in the sense, there is no exine on that part. And that part with no exine is called germ pores, through which pollen tube usually emerges. It can be 3 germ pores in dicot, 1 in monocot. So there is a term for it, tricolpate, monocolpate, based on number of germ pores where exine part is not seen. NCERT given us a beautiful diagram showing that exine arrangement, all that things. Now pollen grain, usually cytoplasm is surrounded by cell membrane. So sporoderm will be there, inside it cell membrane, inside it cytoplasm. Now pollen allergy, this is another usual area. Many reasons are there for showing allergy. One is pollen. They can cause severe allergy for many individuals. And this will be mainly caused by wind pollinated flowers, where pollen grains will be liberated through wind. And symptoms include allergy will be there, irritations, asthma, bronchitis. And which are the main plants that can cause this pollen allergy? NCERT, famous for exam point of view. Common is Parthenium. Common name is carrot grass. And this plant came to India along with the imported wheat as a contaminant. Later, this Parthenium get established in India. Another plant is Amaranthus. Another one is Chenopodium. According to NCERT, these 3 can cause allergy, pollen allergy. Next is pollen viability. Pollen grains are viable, but their viability time is variable. In graminea members means plants like rice, wheat, grasses, etc., we can find 30 minutes roughly. And it can be extended to months, as in families like Leguminosae, Rosaceae, Solanaceae. These are pure NCERT exam point of view question, theory entrance. So pollen allergy, pollen viability. Now, pollen preservation through pollen banks. Pollen grains can be used for future use. So it has to be preserved. And preservation is usually done in liquid nitrogen where temperature is -196 degrees centigrade. And that low temperature treatment or preservation is called cryopreservation. And many products are extracted from pollen grains. And that will be providing extra energy, stamina, all this can be developed. So pollen tablets are now available. NCERT again given a diagram for it. Now we have to proceed to next heading, from microspore, how male gametes will be formed. So pollen grains or microspores undergo mitosis, because already meiosis also it become haploid. Now only mitosis can take place and gives two cells. One is called vegetative cell and one is called generative cell. It's another area of question. Vegetative cell, it will be larger with irregular shaped nucleus, large amount of food material. Generative cell, it is smaller with a spindle-shaped nucleus. Shape of the nucleus itself is different. And this generative cell will undergo one mitosis to form two non-motile male gametes. So generative cell, smaller with a spindle-shaped nucleus, undergo mitosis. Two non-motile male gametes will be formed. And in majority of angiosperms, pollens will be liberated at two-celled stage. It will be around 60% majority. And remaining 40% will be roughly around 40% will be three-celled stage. That is regarding how microspores pollen grain will be converted into male gametes. So pollen grain form undergo mitosis, vegetative, generative, their features, and generative cell will give rise to two male gametes after mitosis. 60% of angiosperm, normally two-celled stage, it will be liberated, pollen grains, and remaining it will be three-celled stage. So this is a chart showing up to two male gametes. So you can find single meiosis, 4 microspores, then abnormal mitosis, asymmetric spindles, one large vegetative cell and one small generative cell. Generative cell again undergo mitosis to form two male gametes. So one meiosis, two mitosis. Problem type question can be asked. Now, as usual, few theory question, few entrance question along with it, key for the objective type question. After doing it, check the key. Now, once again, we started from the second episode of sexual reproduction flowering plants, mainly dealing with the male reproductive structure. That is how we started today. And you can find and the different parts, stamen, proximal end, terminal end, then what is connective, all this. Then what is the nature of anther, wall layers, lobing, theca. Then microspores undergo meiosis, forming microspores, arrangement of microspores, etc. After that, we discussed about microsporogenesis, a mass of diploid cell, rectangular shape, archisporial cell, transverse division, wall layers, and microspore mother cells. Then pollen grain, their size, layers, importance of exine, importance of indine. Exine is sporopollenin, indine is pectocellose. Then exine sculpturing, ornamentation. After that, cryopreservation, pollen banks, pollen viability, pollen allergy. Then finally, some questions. That is regarding microsporogenesis and microgametogenesis. So if you, after hearing and watching all this, if you like it, subscribe, share, comment, and like it. Thank you. So we'll be meeting with another one, female side. May take 2 episodes because it is a bit big. Possible in 1 episode, I'll try to finish female part. If it is not possible, in 2 episodes. Okay. So we can meet later. Thank you.